Good afternoon, class. It's 3 p.m. and welcome to practice problem set week session for lecture corresponding to week 8. We'll be solving some exemplar questions from axis specification of Drosophila as per the course curriculum for the course Introduction to Developmental Biology. So Professor Subramaniam is taking these lectures following to lecture 8 about axis specification of Drosophila. We'll study about where he teaches about gap genes, pair root genes, segment polarity genes, Hox genes, and the body plan and segmentation in Drosophila. So we'll be dealing with some of the questions here, and as I can see there are not many students here. I'll wait for 5 more minutes and keep admitting students who are requesting to join this call. Meanwhile, a common set of instructions for all the students. Please turn off your mic and video chat throughout the session. The session will be an interactive session and you can interact with me via the chat box. So the way we proceed through this practice problem session is I'll pose certain set of questions equivalent to what you receive in your quiz, and then one by one we'll solve these questions. I'll ask you for giving me the correct answer or guessing the answer. You can type your answer in the chat box, and then later on, after we collect some answers, I'll reveal the correct answer and also we'll understand the rationale behind the correct answer. With that much information, let's wait for 5 more minutes so that more students can join this call and attend this practice problem session. So we'll start sharply at 3:05. We are already 5 minutes past 3, so I'll start sharing my screen and we'll move ahead with the session. Since there are not many students today, I'll keep admitting students who are requesting to join this call. Meanwhile, we are starting this session. So let me just quickly share my screen with the class. I hope you can see my slides, and the slides are changing also. If my audio is clear to you guys, just let me know in the chat with a yes or no whether you can see my slides, the audio is audible, and you can see the slides changing. Good. So let's begin with today's session. So as I mentioned earlier, we will be practicing some problems from lectures of week 8, where Professor Subramaniam is teaching about the axis specification in Drosophila in the course Introduction to Developmental Biology. And my name is Ashutosh, and I'll be your teaching assistant for this course. So the same format we'll follow as we were following in the earlier classes. We'll have a set of 10 questions, multiple choice. Multiple answers may be correct, and I'll pose those questions and ask the participants to answer the questions. It's okay if you do not know the correct answer. You can just guess based on your hunch and understanding, and I'll reveal the correct answer. Then we'll move on to the explanation of the concept behind the correct answer. Cool. And please mention your answer in the chat box for smooth conduction of this session. So the first question we have here is a simple question. Why are GAP genes named so? Why are GAP genes named so? And the options are: these genes activate the GAP protein. These genes cause large gaps in the developing embryo. These activate many apoptotic proteins. Mutations in these genes cause large gaps in the developing embryo. So these are your 4 options. You have to identify why GAP genes are named so. Right? Let's wait for a minute and collect some answers. Simontini, since you are here, you can mention your answer in the chat box. Read the question carefully and let me know what you think is the correct answer. Okay, I can see your answer. You think D. Why are GAP genes named so? Mutations in these genes cause large gaps in the developing embryo. Okay. Now let me reveal what is the correct answer. You are absolutely correct, Simontini. D is the correct answer to this question. Gap genes are named so because mutations in such genes causes large gaps in the developing embryo. So let me give you a fun story behind it. In earlier days, during Drosophila, which was the era of Drosophila genetics, these Drosophila scientists had a funny notion of naming Drosophila genes, but also a scientific viewpoint to that funny notion. The genes were named after the mutant phenotype that was observed in the flies. So since the scientists observed gap-like mutations, certain regions of the embryo, large sections of the embryo were missing, causing huge gaps in the body plan. So first the mutation or the mutant phenotype was observed, then the gene was found out. So those genes were named as gap genes. Similarly, we have other genes such as wingless. Wingless codes for a protein which is when mutated causes a wingless phenotype in the fly. That's why the gene is named wingless. A mutation or a loss of function of wingless will cause no wings in the fly. Similarly, loss of function of gap genes or mutations in these genes cause large gaps in the developing embryo. So you are absolutely correct. Now if you look at, this is a schematic showing the expression profile of certain gap genes. So we have example of two gap genes here, CRUPL and NERPS. So CRUPL, as you see, is expressed in all these big regions. So this brown shaded region is the expression profile of CRUPL, and NERPS, whereas overlaps with certain parts of CRUPL expression, but still all these genes code for heavy or large body segments of the organism. So hence when we have mutation, we have these shortened phenotypes that we see here. Other examples, expression profile here, hunchback expression, CRUPL and NERPS. So this is what we would have when we have a mutation in the gap genes. So early embryo, if this is the area of gene action, which is the expression profile of the gap gene, later in the embryo, this area will lead to formation of many segments, formation of many body plans. And in the larva, we see that this body plan is extended and they have a higher area of influence. Now if we have a mutation in such a gap gene, then all this purple region will be missing, where we have, in this case, on denticular preparation, we can see it has lower three denticles fused with the topmost denticle, but there is no sign of these segments which were actually as the domain of expression of the gap gene. So scientists first identified these kind of mutant phenotypes, and later when they found out, okay, these are the genes that are responsible for such mutations, such phenotype, they named them gap genes. So you were absolutely correct, and I hope the explanation and the examples for this question is clear to you. Is it clear to you? Just let me know via the chat. Great, thank you. Okay, let's move on to the second question. Which of the following statements are true? So there are a set of 4 statements. There can be multiple correct options. Which of the following statements are true? Option A, segment polarity genes activate pair rule genes. Option B, pair rule genes activate segment polarity genes. Option C, pair rule genes act on a particular part of every segment. Option D, pair rule genes only act on some segments. Now out of these statements, which are the statements that are true? Take your time and let me know what do you think is the correct answer. And if you're not able to figure it out, you can just guess or let me know that you're not able to figure it out. Then we'll move forward. One. Okay, I see you have answered. Statement 1 you think is the correct answer, right? Okay. Now let me reveal what is the correct answer to this question. Statement 2 is the correct statement. Pair rule genes activate segment polarity genes. So pair rule genes are the upstream player. Segment polarity genes are the downstream player. And statement 3 is also correct. Pair rule genes act on particular part of every segment. So pair rule genes are present in every segment, but they only act in a particular part of every segment. While the other two statements, statement 1 and 4, are not true. Segment polarity genes does not activate pair rule genes, and pair rule genes does not only act on some segments, whereas it acts on particular part of every segment. So these two statements will be the correct answer. Now let us understand what is the hierarchy between pair rule genes, segment polarity genes, and all the axis specification, all the zygotic genes that come into play after gap genes, right? So in a Drosophila egg, when the egg is still in syncytial blastoderm form, means there are multiple nuclei with a common cytoplasm in the Drosophila egg. In that stage, there are some genes which are in effect, which are known as maternal effect genes. So these maternal effect genes are the ones that are passed down from the mother to the organism. The mother, when it lays the eggs, it deposits some genes inside the egg when it's in the syncytial form, and those genes are the maternal effect genes. So in the Drosophila axis formation, the first gene that comes into picture are the maternal effect genes. Then comes certain zygotic genes which activate after fertilization, right? So maternal genes determine the anterior posterior axis and induce the gap genes. So what these maternal effect genes would do, they would turn on instead a set of another set of genes which are very important, called gap genes, which we encountered in the first question. So gap genes define several broad areas and regulate pair rule genes. So if you see the expression of maternal effect genes, an example is bicoid and nanos, which form gradient from anterior, defining the anterior and the posterior region in the syncytial blastoderm. When you see the expression of gap genes, you will see broad areas of expression defining certain gap genes, as you can see on the left hand side figure. Gap genes define several broad areas and regulate other genes called pair rule genes, which define segment locations. So pair rule genes, you can see, have shorter expression, and you can divide the Drosophila body into different segments. So pair rule genes have alternative segment expression. Then comes segment polarity genes. Pair rule genes regulate segment polarity genes, which determine the boundaries of anterior posterior orientation of each segment. So within each segment, as you can see here, this each gray square is a segment. Within that, the green expression is towards the anterior side. So this is a segment polarity gene which determines the anterior posterior polarity within a segment, within a single segment. And together, in the presence of gap genes, pair rule genes, and segment polarity genes, the most downstream player that comes into action is the Hox gene. So this is the hierarchy of gene activation in Drosophila axis specification and patterning. So maternal effect genes will lead to gap genes control pair rule genes control segment polarity genes, and all these three, gap, pair rule, and segment polarity, together control Hox genes. So statement 2 will be correct, which we saw. Pair rule genes activate segment polarity, not the other way around where segment polarity activate the pair rule. Now let us study a little more about pair rule genes, because our third statement, which is the correct answer, describes about expression pattern of the pair rule genes. Pair rule genes are expressed just before cellularization in patterns of 7 stripes, half the number of segments in a wild type larva. So here you see pair rule genes are expressed just before cellularization, just before the syncytial blastoderm ceases to exist. Pair rule genes are expressed, and segment polarity genes, which are further downstream of pair rule genes. Now segment polarity genes will express in the cellular stage, while maternal effect genes, gap genes and pair rule genes will still be expressed in the syncytial blastoderm. Some pair rule genes respond to patterns already laid down by the maternal and gap genes. So as we know, the top order genes such as maternal gap genes lay the foundation groundwork for working of pair rule genes, while others are regulated by pair rule genes. Pair rule gene expression is very dynamic. 7 stripes. So if there are a total of 14 stripes, pair rule genes will express in every alternate stripe. Here is an example here. Even skipped and Fushitarazu are two pair rule genes which express in alternate stripes. And these are turned on by maternal genes, gap genes. So that's why the third option will be correct. Pair rule genes act on a particular part of every segment. Of every segment, it will act on a particular part. It might not be the same pair rule gene, but two different pair rule genes but they have expression in every segment. That's why statement 2 and statement 3 are the correct answer. I hope you are clear, Simantini. If so, then let me know. Great. Okay. Moving on to the next question. I think you will be able to answer this. It's again a true-false kind of question. Which of the following are false? Statement 1. When the segment polarity genes are activated, the embryo exists as a syncytium. When the segment polarity genes are activated, the embryo is cellularized. Statement 3. Highest concentration of bicoid is required for the formation of anterior most structures. And option D. Hunchback is activated when the concentration of bicoid is slightly lower than in the anterior most part. And statement 5, Fushitarazu is a gap gene. Now out of these 5 statements, which of the following are false statements? Mind you that there might be more than one answers to this question, as you must have encountered in some quizzes from Professor Subramaniam himself. So 2 of the statements are regarding segment polarity genes, 2 other statements regarding bicoid, and the last statement is regarding Fushitarazu is a gap gene. Take your time, read through all these statements and let me know what you think is the correct answer. Okay, I see your chat box, Simantini. One of them will be option 1. You say, when the segment polarity genes are activated, the embryo exists as a syncytium. Okay, I'm not sure about the other. Fine, it's fine. Good job. Okay, let me reveal the correct answers to this question. Very correctly, as you said, you are very correct. Option 1 is the correct answer. This is a false statement. Segment polarity genes are activated when the embryo is cellularized. Just in the previous question, I gave a hint for the answer. Maternal effect genes, gap genes, as well as pair rule genes express when the embryo is still in syncytium. But segment polarity genes, Hox genes, come up when the embryo is cellularized. So you just have to remember the order of hierarchy and then you're good to go. Okay. Now the next statement, which is false, is statement number 5. Fushitarazu is a gap gene, is a false statement. Fushitarazu is an example of pair rule genes. I think we just saw in the earlier question itself, even skipped and Fushitarazu, for your information, are examples, well studied examples of pair rule genes. While Krupal, Giant, NERPs, these are gap genes. Fushitarazu is a pair rule gene. Okay. Now let us have an understanding of all these statements through our concepts. As I was mentioning, what are segment polarity genes? Segment polarity genes in Drosophila are expressed in individual cells, not in syncytium. So this is the statement that we were referring to. These genes play crucial role in establishing, maintaining the polarity of individual segments within the developing embryo. So when we have pair rule genes, now you see there are different segments here mentioned as T1, T2, T3, A1, A2, A3. If you can follow the diagram and my pointer, within each of these segments we have two genes, engrailed and wingless. These genes determine the anterior posterior axis within each segment. So if you can divide the body into various segments, each segment will have its own anterior posterior axis determined by segment polarity genes. Segment polarity genes are part of larger gene regulatory network responsible for segmentation patterning during embryonic development. So together with pair rule genes, gap genes, segment polarity genes and Hox genes, the whole body plan and patterning from anterior to posterior axis as well as dorsal to ventral axis is laid down via combination of these different genes. So again, emphasizing here in this figure is the order of hierarchy: maternal effect genes, then comes the gap genes, then comes the pair rule genes, finally segment polarity genes, and all three are involved in controlling the Hox genes. And finally we have a segmented body plan of the Drosophila larva. T1, T2, T3 form the thorax, A1, A2, A3, A5, A6, A7, A8 forms the abdomen. So here is an example of segment polarity genes. Statement 1, that is why we have correct. Segment polarity genes as activated, the embryo exists as a cellularization. So this statement is false. Now let us look at the other two statements where it was, the statements were highest concentration of bicoid is required for formation of anterior most structures, and hunchback is activated when concentration of bicoid is slightly lower than the anterior most part. If you see on my left, these are oocyte mRNAs. Oocyte mRNAs refer to maternal effect mRNAs. And there are several important maternal effect mRNA such as bicoid mRNA, nanos mRNA. Bicoid mRNA localizes to the anterior portion of the egg, while nanos mRNA localizes to the posterior end. And we also have high levels of hunchback mRNA and caudal mRNA. Early cleavage embryo proteins. So this happens when this mRNA is translated into protein. Now in the presence of bicoid protein, hunchback translation can begin. So in the anterior most region where we have high concentration of bicoid, we do not have hunchback expression because that region is restricted for acron. As you can see here, acron forming region, while in the head forming region we have hunchback expression. So we have bicoid mRNA here, localized here, nanos mRNA to the posterior. After fertilization and egg laying, there is a gradient of bicoid protein and a gradient of nanos protein which determines different regions. So the head forming region will express hunchback protein, and expression of bicoid is necessary for determining the head forming region. Whereas in the posterior, nanos protein does not let translation of hunchback. So we have this kind of gradient. If you see, we have hunchback bicoid gradient in the anterior, whereas caudal nanos gradient in the posterior. So that is why the statement 3 and 4 are true. And the final statement about Fushitarazu is a gap gene. Fushitarazu is not a gap gene, it is a pair rule gene. the first indication of segmentation. So let me take you to a brief overview of how does a segment and a parasegment look like. So we have different segments here. Then each segment can be divided from the middle with posterior of one segment and anterior of the adjacent segment. This is known as a parasegment. And here you can see Fushitarazu is an pair rule gene expressed in alternate parasegments. And along with Fushitarazu, there is another there is another gene called even skipped, which skips these even parasegments. So we see here Fushitarazu is expressing in parasegment number 2, parasegment number 4, 6, 8, 10, such like that. So even skipped is named as such because it does not express in even number of parasegments. So it will express in 1, 3, 5, 6. So here we see pair rule genes express in alternating segments as well as in alternating parasegments. So Fushitarazu is an example of pair rule gene. Okay, so if you see here even skipped and Fushitarazu, this is a expression profile here. We can see alternating stripes of red and black. Okay, I think you are clear. I can see your chat. Very good. Thank you. Okay, you are clear. Then we move on to the next question. Which of these are false regarding Claus-Sanders 1975 experiment? So this is an important experiment that even Professor Subramaniam has mentioned, and you can find it in the book. Claus-Sanders experiment, if you remember what it is. So these are the options. It proved that there are two organizing centers that direct the anterior-posterior axis formation. Statement 2, when a ligature was created in the middle, only middle segments were affected. Statement 3, when a ligature was created in the middle, only the posterior segment was affected. And statement 4, when the ligature was created in later stages, the number of missing segments were reduced. Now you have to identify which of these statements are false regarding Claus-Sanders experiment. Okay, I can see your answer already. You are saying statement 1 and statement 3. It proves that there are two organizing centers that direct the anterior posterior axis formation. And number 3, when ligature was created in the middle, only posterior segment was affected. Okay. So the first correct answer, when a ligature was created in the middle, so option 3 is correct. And option 3 is the only correct answer, because Claus-Sanders experiment actually did prove that there are two organizing centers that direct AP axis formation. So this is not a false statement, Simontini. Statement 3 is a false statement, but statement 1 is a true statement. So if you see Claus-Sanders 1975 postulated that two organizing areas form two gradients, one initiated at the anterior end and the other at the posterior end. So what he did, he ligated the early egg in the development from the middle. One half became the anterior end of the embryo and the other posterior half, the middle segments were missing. So when he tied these this in the middle of the embryo, what happened actually was the gradient that is formed at the did not form correctly. So one half became the anterior end of the embryo, fine, because the ligature was created in the middle, the anterior segment was formed. But since the other half did not receive the anterior signals or anterior gradient due to the ligature, the other half had some middle segments missing. So this was Claus-Sanders experiment. Each of these gradients form its own structures at the poles and interact with other gradient to form central portion of the embryo. So this was the finding of Claus-Sanders. So it refers to bicoid and nanos. If you imagine, each of these proteins form their respective structures at the ends. Bicoid will form the head and nanos will form the posterior. But in the middle, different gradients of these proteins lead to development of middle structures. That's why we had these middle segments missing when the ligature was made. Sander based this model on experiments that involved ligating the embryo at various times during development and transplanting regions of polar cytoplasm. First, he moved cytoplasm from posterior pole more anteriorly. He obtained a small embryo anterior to the posterior pole plasm. So first what he did before ligating experiments, he transferred the cytoplasm in the posterior region to the anterior region. So then he saw small embryo anterior to posterior pole plasm while extra segments not organized into an embryo formed behind it. So wherever he implanted the cytoplasm, that small region became AP axis, but beyond that, the downwards, there was no proper organization. Second, if he ligated the egg early in development, separating anterior from posterior, one half developed into anterior but the other half developed into the posterior, but neither half contained the middle segments. The middle segments were absent because now due to the ligature there is no interaction between these different gradients. So that's why statement 1, 2 and 3 are true statements. His experiment proved that there are two organizing centers. When ligature was made, only middle segments were affected, and in later stages the number of missing segments were reduced because in later stages already the interaction has turned on certain expression of genes which can lead to formation of the middle segments. So lesser number of middle segments were missing. I hope this is clear to you. Please let me know. Very good, very good. Cool. Moving on to the fifth question. Why is the hunchback protein not present in the posterior even though hunchback mRNA is uniformly distributed all over? This is an interesting question. Why is the hunchback protein not present in the posterior even though hunchback mRNA is uniformly distributed all over? And the options are: Nanos inhibits hunchback translation at the posterior, caudal inhibits hunchback translation at the posterior, hunchback translation requires bicoid which is present only at the anterior, Nanos activates hunchback translation only at the anterior. Read through the question and the options and let me know what you think is the correct answer. Okay, I see that you have mentioned statement 1 to be the correct answer. Nanos inhibits hunchback translation at the posterior. Okay, let me reveal the correct answer to this question. You are absolutely correct. Option 1 is the correct answer. Nanos inhibits hunchback translation at the posterior. Meanwhile, there is a second answer which is also the correct answer. Hunchback translation requires bicoid which is present only at the anterior. If you see, hunchback mRNA is present all over the embryo, but since we have anteriorly expressing bicoid, bicoid can now sit on modular enhancers of the hunchback protein and turn on and bicoid can promote the translation of hunchback, and Nanos inhibits hunchback translation. That is definitely correct. So again here we encountered this graph before. So hunchback mRNA, caudal mRNA are all laid down and present in high levels both in anterior and towards the posterior, but bicoid mRNA and Nanos mRNA are localized to their anterior and posterior ends respectively. According to the bicoid protein gradient, we see a higher hunchback gradient towards the anterior. Similarly, wherever there is higher Nanos gradient, it inhibits the translation of hunchback. So if you see here what happens in the anterior, bicoid mRNA gets translated to bicoid protein in the presence of all these factors, cortex, rhozone and stophin. Now bicoid protein can inhibit caudal mRNA translation to caudal protein. That's why we do not see caudal protein presence in the anterior. While in the posterior, nanos mRNA in the presence of OSCAR converts to nanos protein. Now nanos protein with the help of Pumilio for P55 proteins like this inhibits translation of hunchback mRNA to hunchback protein. That's why we do not see hunchback in the posterior region of the Drosophila. So in a summary, bicoid at the anterior end translates HB. HB is hunchback while inhibiting caudal. Nanos traps HB mRNA and prevents translation in the posterior end. So these are the two correct answers. Bicoid translate HB while nanos inhibits HB. HB here refers to hunchback. That's why we have a anterior high to posterior low gradient of hunchback protein. I hope this is clear. It's a pretty simple explanation, and you gave the answer correctly, but you just missed one statement. Okay, cool. Moving on to the sixth question. What is the significance of parasegments? What is the significance of parasegments? Statement 1. It coordinates the movement of a segment independent of its adjacent segments. Statement 2. It coordinates the movement of adjacent segments. Statement 3. Alternate parasegment, the same pair rule gene is active. Statement 4. Within each segment, the same pair rule gene is active. Now you have to tell me what is the significance of parasegments. Mind you that there might be multiple options correct. Take some time and let me know what you think is the correct answer. One and 3. Okay. It coordinates the movement of a segment independent of its adjacent segments. It coordinates the movement of adjacent segments. I think now when you answer, sorry. So as you see here, statement 3 is the correct answer. Alternate parasegment, the same pair rule gene is active. And statement 2 also happens to be the correct answer. It coordinates movement of the adjacent segments. Now how? I'll tell you how. We'll understand about it. The control of adjacent segments in Drosophila development primarily involves actions of genes called pair rule genes and segment polarity genes. So these are the two genes which control adjacent segments, which refine and maintain segmental boundaries established by initial parasegmental pattern. These genes create alternating patterns of gene expression that specify anterior-posterior polarity and distinct segmental identities. And parasegments are defined by specific boundaries of gene expression patterns essential for organizing embryo into segments. So once the parasegment is determined, parasegment also determines the boundaries of expression of pair rule genes as well as segment polarity genes. So pair rule genes will occur in every alternate parasegment, and within a single parasegment there will be segment polarity genes. So indirectly, a parasegment has significance on controlling adjacent segments, since we have repetitive expression of pair rule genes as well as segment polarity genes. So parasegments serve as transient repeating pattern or template that helps establish segmental boundaries during early embryonic development. So we can in a form say that parasegments are significant for controlling adjacent segments or movement of adjacent segments in the way that they define the expression of different pair rule genes as well as segment polarity genes. And for the second statement, which was actually correct, here is an example of even skipped and Fushitarazu, as I was mentioning earlier in the questions. Alternate parasegments have the same pair rule gene. So if you see here, 1, 3, 5, 7 alternate parasegments have even skipped, which has skipped the even parasegments. Fushitarazu is present in 2, 4, 6, 8. So alternate parasegments, same pair rule gene, while same parasegments, different pair rule gene. So you were correct, and I hope this is clear. Pretty simple, straightforward explanations, but still, if you have any doubt, you can ask me. Shall we move to the next question? You just let me know. Very good, very good. We are moving quite fast today. We might end our class a little early. So it is a 2-hour time slot, and whenever we end within it will be the end of the class. So here we are at question number 7. Which of the following are true? Again, true false statements. Actually, Professor Subramaniam loves these kind of confusing true false statements. So you have to be very careful with your concepts and when you answer them in the real exam, right? Option 1, bicoid and hunchback at the anterior side activate giant. Option 2, giant activates crupple. Option 3, hunchback activates nerves. Option 4, hunchback and nerves are mutually antagonistic. Now, which of these are true? Read through all the options carefully and just let me know what is the correct answer. I'll be waiting in the chat box. Okay, I see you have mentioned option 2, giant activates cripple. Yes, okay. Let us see. I'll reveal the correct answers to this question. Statement 1 is true. Bicoid and hunchback at the anterior side activate giant. And statement 4 is the correct answer. Hunchback and nerves are mutually antagonistic. So these will be the correct answer. So there is a whole lot of networking between all these gap genes that we see here, that I have a diagram of. I'll show you. Giant is a gap gene which is activated by bicoid and hunchback, while hunchback and nerves are mutually antagonistic. So there is a whole lot of relation. You might have to memorize some of that. So it is just a factual based question. So you see here, this is a complex network of different gap genes and the relations between them. So here is our giant protein, GT. So if you see here, bicoid along with hunchback, bicoid will activate giant. So this statement will be true. Bicoid and hunchback at the anterior side activate giant. Giant is activated at the anterior side because of bicoid and also because of hunchback. Giant activates crupple, which is actually not true, whereas giant inhibits crupple. High levels of giant will inhibit crupple and high level of crupple will inhibit giant. So hunchback in a form also plays a role in inhibiting giant through couple. Hunchback can lead to expression of crupple or can lead to inhibition of crupple. Whereas nerves, if you see another gap gene, hunchback activates nerves. Hunchback does not activate nerves, it inhibits nerves. And hunchback and nerves are mutually antagonistic. So this relation holds true. So that's why the statement 1 and 4 are true. Bicord activates giant, hunchback NURBS mutually antagonistic, and cripple and giant are mutually antagonistic. Gap genes are activated in response to maternal morphogen gradients. So remember, the topmost hierarchy gene is the maternal effect genes, which then turn on gap genes. The activation of gap genes follows specific sequence along the anterior-posterior axis. Here I have mentioned different gap genes. Cripple is activated in the central region of the embryo, where levels of bicord and caudal are intermediate. So here if you see high caudal level, like when caudal level starts going up, we can see the expression of giant. But caudal will be inhibited wherever there is high bicoid. So that is why in the anterior region we have less caudal. So we do not have much of giant. But towards the middle areas where we have lesser amount of bicoid, we have some amount of caudal present. Now both the bicoid and caudal can collectively induce expression of giant and cripple. Sorry. Yes. NURBS is activated in the posterior region of the embryo, where bicoid levels are low and caudal levels are high. So in the posterior region it will be NURBS. Here KNI NURBS. Giant is activated in the anterior region embryo, where both bicoid and caudal levels are relatively low. So cripple and giant follow the similar kind of relationship here. Relatively low levels of bicoid and caudal will cause activation of giant. Hunchback, which was initially activated by maternal factors, continues to be expressed in the anterior region of the embryo. Hunchback will be expressed in the anterior region. Tailless and giant are activated in the posterior region of the embryo, where caudal levels are high and bicoid levels are low. So there is another gap gene called tailless. Along with giant, it is also expressed in the posterior region. So NURBS to the posterior, hunchback to the anterior, cripple to the intermediate or middle sections, giant to the middle and the posterior. I hope this is clear. You just have to look at this interplay of networks and might be remember few of these gap genes. You might get 1 or 2 questions. It's not a very difficult thing to understand. You just have to memorize it sometimes, you know. Okay, you understood. Very good. Moving on to the 8th question. Which of the following are false regarding pair rule genes? Again, a true-false based question. Which of the following are false regarding pair rule genes? Their enhancers are modular. They are regulated by different concentrations of gap gene products. Expressed in a narrow band compared to gap genes, expressed in a broad band compared to gap genes. Some are activated early while some activated later. So these are all the statements. Go through them and tell me which of these statements is a false statement regarding pair rule genes. I see you have mentioned your answer. Statement 4 is false, is what you say. Okay, I'll reveal the correct answer now. Very good, absolutely correct. Statement 4 is the correct option. Pair rule genes are not expressed in a broad band compared to gap genes. Rather, gap genes are expressed in a broader band comparison to pair rule genes. Very good. So we'll go through the statements one by one. The first statement was modular enhancers in pair rule genes. So here we have example of EVE, which is even skipped. So you see what do I mean by modular enhancers? The single gene coding region, which is mentioned in red here, reddish orange, produces the even skipped mRNA. But to control that, the same gene has so many enhancers, and particular set of enhancer will only express the gene in particular set or segment. So stripes 3 and 7 is activated by this enhancer, stripe 2 by this enhancer. Muscle precursor cells, if you see in stripe 5, stripe 1 and 5 will be activated via this enhancer. So there are many enhancers, many different specific enhancers which can control the expression of gene. That is why when we selectively activate even skipped with a tissue specific or stripe specific enhancer, we can see just the activity of even skip protein in those stripes. Thereby we say that all these genes, pair rule genes, segment polarity genes, have modularity in their enhancers. EVE gene enhancers show modularity. Stripe specific enhancers in the EVE locus, a map shows the position of enhancers. Again here, this shows that stripe 5 enhancer here, stripe 1 enhancer, 4, 6 enhancer, 2, 3, 7, and there are also late enhancers, so which express in the late stage. Simple patterns of 1 or 2 stripes of reporter gene expression are driven 5 discrete enhancers. So there are discrete enhancers. The initial patterns of 7 EVE stripes represent some of the activity of these enhancers. This is a verdict for modularity of the enhancer. A separate enhancer directs 7 stripe pattern that appears after the initial pattern is established. So this now confirms some of the pair rule genes can occur early in time and later in time. This enhancer is important for maintaining and refining the pattern. Also in the earlier question where we were dealing with pair rule genes, there are examples of some early expressing pair rule genes and late expressing pair rule genes. So even skipped is a early expressing pair rule gene, while Fushitarazu is a second or late expressing pair rule gene. I hope that is clear to you and you have answered very correctly. We are moving on to the 9th question. Which of the following are false? Again a true false question. Homeotic genes are regulated by gap genes and pair rule genes. The arrangement of homeotic genes in a chromosome corresponds to the domain of expression in the body. Homeotic proteins contain the homeodomain, which is the conserved protein sequence. Hox genes are conserved among many organisms. Homeodomain consists of 180 amino acids. So out of these 5 statements, which statement do you think is a false statement? And this is generally about Hox genes or homeobox genes. Okay, I see you have mentioned option 1 as the correct answer. Homeotic genes are regulated by gap genes and pair rule genes. This is a false statement. I would like you to reconsider your options because we just studied about what is the hierarchy. If you remember the hierarchy again, you might want to change your answer. Homeotic genes 5 also. Okay, you are saying statement 5, homeodomain consists of 180 amino acids. Let me reveal the correct answer. You are damn right. Statement 5 is the false statement. Homeodomain consists of 180 amino acids is a false statement, but there is no other statement which is false. Statement 1 is a true statement, Simontini. Because, yeah. Because homeotic genes are regulated by gap and pair rule genes. If so, let us see the hierarchy again. Let's revisit the hierarchy. Again, we have maternal genes, gene group, examples of region of gene activity showing here, and name of the example. So we have an example of maternal gene, dicoid. Zygotic genes then come into play. Gap genes, hunchback, pair rule genes, even skipped, segment polarity genes, engrailed, and all these three, gap genes, pair rule, and segment polarity, help in maintaining homeotic genes such as abdominal A. So this is the hierarchy, and you have to remember it. It is very important. Maternal genes, gap genes, pair rule genes, segment polarity genes, then comes the homeotic genes. Hox genes, also known as homeobox genes, are a family of highly conserved genes found in many animals, including humans. So if you check all these animals, starting from invertebrates, the Drosophila organism is an invertebrate organism, as well as in humans to vertebrates, Hox genes will be found everywhere. They are conserved, highly conserved set of proteins. They play a fundamental role in controlling development of an organism's body plan and specifying the identity of body segments along anterior to posterior axis. So why they are conserved? Because they are playing such an important role. What role do they play? All these Hox genes are involved in segmentation of the body plan along the anterior posterior axis. So all the organism, irrespective of whether it's a vertebrate or an invertebrate, will have a body segmentation profile, and these Hox gene expression determines these segmentation. That is why they are so important, and that is one of the big reasons why they are highly conserved across all these species. This diagram here shows a comparison between Hox genes of a Drosophila and a human. So in the Drosophila, we have head, thorax, abdomen, three parts, body parts, where the head is towards the anterior side and the abdomen towards the posterior side. Drosophila home C and ancestral home C. If you see homeodomain protein C, we have different genes here: lab, PB, DFD, SCR, antennapedia, ultrabithorax, ABDA, ABDB. So these are different Hox genes. While in the ancestral home C, you can see there is so much sequence similarity and conservation. You can find the genes at the same region in the ancestor as well as in the Drosophila. Now if you compare humans' Hox genes, Hox A, B, C, D, you can find the labial one corresponding having high homology with the A1 gene, PB having high homology with A2, A3, DFD is conserved across all these ABCD Hox groups. Similarly, there are high degree of conservation in protein structure, sequence and function. And similarly, you can see the same anterior to posterior patterning in the human embryo is performed by all these Hox genes from anterior to posterior. That is why they are so important, and that is why they are conserved. And for our answer, homeobox genes contain a 180 base pair DNA sequence that provides instruction for making a string of 60 amino acids protein building blocks known as the homeodomain. So the question was a little confusing because the homeodomain has 180 DNA bases. Now 180 DNA bases will code into 60 amino acids, since one codon will code for one amino acid. Now one codon is comprising 3 nucleotides or 3 base pair of the DNA. So 180 by 3 will be 60 amino acids. This 60 amino acid is the homeodomain. So that statement was false. Homeodomain consists of 60 amino acids. And homeodomain act as transcription factors, bind to DNA, control transcription, and initiate patterns of gene expression. So this homeodomain is conserved among all the members of Hox genes. So this will be the correct answer. This is the false statement. Homeodomain consists of 60 amino acids will be the correct answer. I hope it is clear. Simontini, you have answered very nicely. Statement 5 was the correct answer. Very good, very good. Moving on to the final question, question number 10. What happens when antennapedia is expressed without being inhibited by ultrabithorax? So it is another follow-up question of homeodomain Hox genes. The antennapedia gene expressed without being inhibited by ultrabithorax. What will happen? The fly will have wings even in the third thoracic segment. The fly will have 4 wings. The fly will have 6 legs. Fly will have 2 wings. What will be the phenotype of the fly? You have to think a bit about all these Hox genes and how they are arranged and what structures they are responsible for forming in the Drosophila body. If you express antennapedia and you knock out ultrabithorax or you remove ultrabithorax, then what would happen? Simantini, are you able to figure it out that what would be the resultant phenotype? Antennapedia is expressing but ultrabithorax is not there. If you are able to figure it out, you can tell me the answer, or you can guess, or just let me know, then I'll reveal the correct answers. Okay, I see that you are not sure about this answer and you are having some network issues. It's fine, we are coming to the end of this session. I'll quickly explain to you the last question and we are good to go. Okay, so what will happen when antennapedia is expressed without being inhibited by ultrabithorax? The fly will have wings even in the third thoracic segment. If you remember, there are 3 thoracic segments, T1, T2 and T3. So even in the third thoracic segments, so generally the second thoracic segment is the one which forms wings. The fly will have 4 wings because now it has an extra pair of wing in the third thoracic segment. The fly will have 6 legs because 6 legs is the general phenotype. The fly will have 2 wings. This will not be the answer because the fly will have 4 wings, 6 legs and wings in the third thoracic segment. Now here is a representation of different Hox genes responsible for the formation of structures in Drosophila. We have an antennapedia cluster, we have bithorax cluster. Here the genes of interest are antennapedia, the green one, and ultrabithorax, UBX, the blue one. So if you see here, thorax from T1 to T3, we have excretion of SCR. Antennapedia marks the second thoracic segment and the third thoracic segment is marked by ultrabithorax, right? Now if there is no ultrabithorax, there will be expression of antennapedia. There is no inhibition of ultrabithorax to antennapedia. Now that the inhibition is removed, antennapedia will start expressing in the T3 segment. So in the third segment we will have 2 more wings and 2 legs. So the body of normal adult fly contains 3 thoracic segments, all of which produce pair of legs. The first thoracic segment does not produce any further appendages, but the second thoracic segment produces both a set of legs and a set of wings. The third thoracic segment produces a set of wings and a set of balancer known as halters. So we'll have 4 wings. The third thoracic segment will have wing. We will have 2 more wings in the T2. We'll have 2 more wings in the T3. And then in the first one, we'll have just a pair of legs. But now since we have antennapedia expressing in the T3, we'll have 2 more legs. So we have 6 legs. So when ultrabithorax gene is deleted, the third thoracic segment, characterized by halteres, become transformed into another second thoracic segment. The result is a fly with 4 wings. So the first thoracic segment, second thoracic segment will be more like the third thoracic T3 will convert into T2 due to expression of antennapedia and absence of ultrabithorax. So it will have 2 more wings and 2 more legs, which was earlier T3 where we had halteres, right? So we have 6 legs and 4 wings. That will be the phenotype. Yes, I think you have understood it well. And with this, we have come to the end of our session. I'll just stop presenting my screen. Yeah. So with this, we have come to the end of our practice problem session in week 8. So next Saturday, we'll be solving week 9 lectures. I can see there are not many students today, and it is the case for most of the classes. But irrespective of the student population, I am obliged to do my job. I'll keep solving these questions, and this is a recorded lecture, so you can avail it on YouTube and access to all those students who are not able to attend my class can go to the YouTube and do it. Anyways, I have to keep teaching, and thanks for attending this session. I hope some of your doubts are clearer now, and you are able to understand the subject better. That's what my job is, to help you solve these questions and help you better. Okay, cool. I'll end this call now and see you on next Saturday. Till then, bye bye. Thank you.